So I'm going to go ahead and share my screen, and I think what I'm going to do that's a little bit different than some of the other folks have done is I'm going to talk certainly about APLI, but what I'm also going to do is try and put it in the context of how we're approaching the COVID-19 problem. Like many other companies, we have a number of directions, and I think what may be a little bit different is to try and show you what our approach has been to understanding the scope of the problem and then how to address it. And so, as you said, APLI is really about the global fight against infectious diseases, and there's our disclaimer and forward-looking statement as a public company. Our lawyers will now be happy with me. And I think it's worth just mentioning that the critical need for antimicrobial R&D is not lost on us, but at APLI it certainly has been a driving force beyond COVID-19. And I think antimicrobial infections are on pace to grow and will surpass deaths worldwide from cancer. And I think that's certainly something that we are focused on, not only here, but beyond COVID-19. And so really what we do that's a little bit different is we're not grounded in any particular technology. We are always trying to tackle whatever global infectious diseases in front of us based on this idea that we're a portfolio company. We were founded by an investment bank, and in full disclosure, I'm a member, a founding partner at Bloom Burton & Co. And so we are very transactional. We build a diversified pipeline of programs based on not only my experience, but the team's experience. And, those are certainly listed here. And you'll notice I'm not highlighting COVID-19 specifically, but we are addressing those needs. And so the question then becomes, how do we, as a number of people who are trained in the space, myself as a physician and a scientist in infectious diseases, a number of PhDs on our team, we approach these issues with a scientific bent, but this idea again of trying to drive a pipeline that addresses problems that we think are tractable. And so one of the things that I'd like to talk about is where we are right now. And I am certainly confused by the level of noise in the system and the amount of news that we're confronted with. And so I thought I'd talk about a couple of different therapeutic options and then walk through an example of how we might address and have been addressing some of the problems in the diagnostic space, because I think that's also very telling. So REMDESIVIR, for all of those who have heard, is one of a couple of different therapeutic options. How do we treat COVID-19? REMDESIVIR is an investigational drug. It's given intravenously, and it's what's called a broad spectrum antiviral. It's been used a lot. It's one of the products that I had the pleasure of working with when I was at FDA during the Ebola outbreak. And some of these have come back again because they do have broad activity. So when we look at these, we start to look at them scientifically. And I think as folks are trying to figure out where the opportunities in the space may be, I'm hoping to leave you with how we approach some of these products. We look first at the science. How does this thing work? In this case, it terminates RNA transcription. It works against SARS-CoV-2, so it has broad activity. And it works against a number of other coronaviruses, and the trials are out there. What I might add is that, today there was news disseminated that REMDESIVIR didn't work. There was some leaked news. And so how do we know what's important here? And I guess I would highlight that really what we should be looking for in all of these products are randomized controlled trials, large trials that are aimed at understanding whether and when these drugs actually work. To put it in context, REMDESIVIR, Gilead product, great in vitro evidence of activity, but it didn't work against Ebola. And so we didn't know that until we ran the trials. And so I guess I would caution, as we look at all of these products, let's keep in mind that the gold standard, the randomized controlled trial, is really what we should be looking for. There really are no shortcuts. And when we take those shortcuts, we run the risk of really getting it wrong. And so with that, I'll jump to another example here, one that's in the news a lot. It's certainly got a lot of play from politicians and others claiming that hydroxychloroquine or chloroquine is the answer. And I think the jury is still out, but I think we're starting to gather more information that hydroxychloroquine not only may not work, it certainly doesn't seem to work in the sickest patients based on the clinical trials that have been run. But it also has a number of safety problems. And one of those is something called QT prolongation or heart rhythm or rhythm disorders. Anyone who's taken chloroquine, for example, it also knows as an antimalarial, you have very, very vivid dreams and other side effects, safety effects as well. Hydroxychloroquine and chloroquine, they're oral, which is great. They are used, the trade name is Plaquenil, used for certain inflammatory conditions and autoimmune disorders. But again, we have to do the trials in order to know whether these things work. And so we spend a lot of time with our scientific team looking for opportunities by first starting with the science and then moving into whether or not these things work in a randomized controlled trial, large trials that are designed to really look at efficacy as well as safety. And so I would say that as all of the listeners are out there looking for potential new products, ask those questions. We certainly ask those questions of scientists all the time. Where are the data that suggests this works in a large informed clinical trial? And I think that will start to point us in the right direction. It's not the quick or easy way to do things. It takes a lot of work, but I think it's certainly worthwhile. So I wanted to spend a little bit of time talking about diagnostic tests. And I have to say, very early on in the process, I asked our scientific team to go ahead and take a look at the tests that were available on the market, because I could see that not only those early tests, those RNA-based, DNA-based and RNA-based tests were valuable to see who was sick, but also the post-illness tests, those antibody tests. And so I wanted to talk a little bit about that and why I think that's important, both from a public health standpoint, and then also as, again, people are looking for opportunities and companies that are making all kinds of claims, how we might be able to evaluate the usefulness of those tests. When I asked the team to go and take a look, we found no fewer than 40 antibody-based tests, primarily coming out of other countries, where folks were making claims about their efficacy, saying it was 90% sensitive or specific. And so what does all of that mean in the context of how these tests work? To first look at why testing, I think I put up here on the left-hand side a snapshot of early COVID-19 testing per capita. And what you'll see is that in the United States, we did a pretty poor job of testing a large population. And I think there's a direct relationship between the number of tests per million people or per capita and the eventual outbreak. The United States, as of March 11th, had done 7,700 tests approximately. We have certainly the most number of cases in the world, and I would argue that we are probably underreporting a lot of those cases. In contrast, South Korea was very, very aggressive about their testing, and I think they were able to then test, trace, figure out who was sick and who was around them, quarantine, and then do some follow-up testing. So I have to say the testing becomes very, very important. So understanding what those tests are is also an important factor in helping to control the disease. And so if we look at the testing windows that are out there, I think it's informative to look at where the tests are and where they sit during these windows. Early on in COVID-19, you certainly have a number you have an asymptomatic stage, which is quite different than some other diseases. Probably 20 to 30% of folks are asymptomatic based on the data that I saw in the last couple of days, depending on where you are. You then have onset of symptoms. and then you have a full-blown illness, and then you have a recovery period. And so I would argue that we can break the testing windows up into two big areas, that that asymptomatic and onset of symptom stage. When can we discover that someone is actually has replicating virus that can be transmitted to others? And that's that blue curve that we see in the chart. And those are RNA and antigen-based tests that we saw very early on in the process, and they're very sensitive and specific, but they're lab-based tests for the most part early on. Some have flipped a little bit, and that allows us to determine who's sick and who's likely to spread the virus to others. As we are exposed to virus, our immune system becomes activated. And we have a specific immunoglobulin or immune system molecule that becomes detectable. That's IgM. That's an antibody. And then we have a longer-lived IgG antibody that remains in the blood and provides long-term immunity. And so those are those IgM, IgG tests that you see for those antibody tests. And the importance there is that those antibody levels tell you that while you may be outside of that area where you're actually sick and spreading virus, it will tell you that you have been exposed previously. Once you've been exposed, the thought is to be determined that you have some sort of immunity from getting sick again, and that will allow us, of course, to get back to what may be more normal functioning economically. I would caution that we don't actually know what the level is that provides long-term immunity yet. But it is a measure, and those are those antibody tests. A lot of these tests go on to claim that they're very sensitive and specific, and they use terms like a positive predictive value. How likely is the test to demonstrate that if you test positive that you actually have that you have been exposed, and the negative predictive value, the opposite of that. And so I would say, why is that important? It's important to know that those numbers actually mean something. And so let's just assume for a second, for a practical example, let's look at how positive predictive value works, because a lot of these tests label it as such. And so that becomes an important function of how these tests work. And so if you have a workforce of 100 staff to test to determine who's been exposed and who can go back to work, and the true unknown prevalence of COVID-19 in your workforce is only 5%. So you have a pretty good understanding of the prevalence, how common the disease is. 5 unknown staff have the illness and 95 have not. And if we apply that test to the population, all of the staff, 5 who have had the illness are tested. And since a test claims that it has close to 90% sensitivity, commonly held out number, again, I saw that on probably 30 or 40 tests that we could get from overseas. 4 people test positive, 95 who haven't had the illness are tested, but with a false positive rate of almost 10%, another 9 people test positive. And so what does that mean? That means that of your staff, 13 people tested positive as having had the illness, but in truth, only 4 of the 13, 30%, have actually had the illness. And so these have real consequences for determining who can go back to work. And as we said before, those people who tested positive but have not had the illness may think that they're immune from getting sick in the future, when in fact they're not. And I think that's a really, really important function of these numbers. We've since learned that a lot of the tests coming from overseas claim that they are almost 90% sensitive. It turns out sometimes they're 20 or 30% sensitive. There's real quality problems. And so before we jump on those numbers, they've got to be tested. We've got to understand what that means. And if we're not careful, we could be exposing a whole bunch of people who may think that they're immune from the disease from getting to getting sick again. And of course, then that's where we get that kind of double rebound. So these are some of the approaches that we take at APLI when we look for opportunities. And I would argue that as you look out at the landscape of therapeutics and testing agents that are out there, we should be applying the same scientific rigor in the middle of the pandemic to our investment opportunities. Science should in fact drive our decision making for these things, and it's certainly really important. And so for us, what we've demonstrated is things aren't as simple as they appear when you read a headline quoting a test with 90% accuracy. We had many people claiming to wanting to sell us tests or license tests to us with a 90% accuracy. But when we did the work and looked at the opportunities and looked at the population with a low prevalence of illness, a false positive can overwhelm the true positives, as we just saw, and make that test less useful. So we don't, as a company, want to endorse or pull in a technology that is going to not help the problem. And if we apply this scientific reasoning to populations with a higher prevalence of illness, such as workers who have had symptoms and are self-isolated, the usefulness of this positive test result increases. And so I guess, not particularly new advice to investors out there looking to assist and help companies with their technology, but we really are in a moment of caveat emptor, buyer beware. Do the work and try and leverage that work to make good decisions.